Day 16, a quick one. We're gonna do 16. I gotta go pick up my daughter soon. But we're still doing it. We're still doing it. And the first things I want to do today is actually figure out I need a new chat. I'm not sure if I do research or not. Let's do a project first and then I do research. So I'm wondering, do we know how the plants track in a seasons? Maybe also specifically day and night? Probably by light. That's my assumptions, right? Should be some sensors like pineal glands for lizard and us to track a light and release melatonin. In plants, there should be also a sensor to track a light. That way they can track the days. But how do they track the seasons? I always thought it's by average temperature dropped or every temperature rise. But also duration is strange. They definitely don't have sensors for time. But do they have a sensor for the whole year? And if they do we have a way to, is there way experiments to trick those sensors? For example, expose them to day and night way faster than regular day and night? Or expose them to the seasons, artificial seasons, way faster than the regular seasons goes? Would that affect the growth? We'll do research, but it's interesting because I wonder if we can do experiments. Basically, the whole claim that you're right, that's about the light, but the story is more interesting than a simple light sensor. And your intuition about temperature is partially right too, just not in the way you'd expect. Wow, we're gonna learn something today. Plants have multiple photoreceptors, proteins, and they are genuinely elegant. The most important family is phytochromes. Let's see. Dark night slow, active inactive, red light, far red light. Oh, they sense red light from the other galaxies? Come on, resolution. White light, red light, fast darkness, far red light. That's interesting. Oh, that's the proteins. PR and P of R. Drought, high temperature, chilling, freezing, celiacal soil, plants, plants disease, and pesticides. So they can sense all of that. Where we are? Right here. which is existing two interconvertible forms. PR absorbs red light 660 nanometer and PFR absorbs far red light 730 nanometers. Where PR absorbs red light, it flips to PFR. When PFR absorbs far red light, it flips back. The key trick: PFR also slowly reverts to PR in the dark, on the order of hours. So the PFR-PR ratio in cell is essentially a running measurement of how long ago the plant lasts. So light, its darkness, timer. Do we have a video of that? 6 Biology Easy Plants.
Phytochromes are pigment molecules of far red and this would be the night.
And
same over here. The day, the peaks represent the day, and the valleys
with music.
We know that plants are living and can respond to sunlight. Photoperiod is the period of time each day during which an organism
This video tutorial is about plant responses, specifically to light and dark. So the question on the slide in front of you: why do poinsettia flower at Christmas whilst strawberries flower in the summer? Hopefully by the end of this video tutorial, you'll be able to answer that question.
Hopefully.
Plants make a phytochrome known as phytochrome red, and that's on the left-hand side of this slide. It's the inactive form of the phytochrome. The phytochrome red is a photoreversible isomer of phytochrome far red, which is the active form on the right-hand side of this slide. Isomers, as hopefully you're already aware, are molecules that have the same molecular formula but have different arrangements of the atoms in space. It's the ratio of phytochrome red to phytochrome far red that enables a plant to determine the length of day and night. In sunlight, phytochrome far red accumulates. Phytochrome far red, the molecular structures of phytochrome red isomer and...
So my question is that ratio, right? It's going from regular to far red. How fast is going, and how can we catalyze that process? If we basically artificially create artificial night and artificial day for the plant, would that process cycles increase? So if we artificially create a dark time and light time, it's actually more rapid than a regular one. Would the cycle of this increase and the ratio also shift more rapidly? Basically, I'm wondering what would be catalyzed for this ratio to go one way or another. If we create artificial cycles that's faster, would that trick the plant thinking that it's day and night, that it's actually faster than normal day and night? Let's say we take a plant into completely dark room, create artificial light, including red light, and far red light, red light, full spectrum artificial light, and turn it on and off 4 times per 24 hours. Not only 2 times. not sure if they understand me, but hopefully it's LLM. We'll see. Day and night, the circadian clock. Plants have a bona fide molecular. Plants also have cryptochromes, blue light sensors, and phototrophins for directional growth toward light. Okay, but phytochrome, the ones that matter most for your questions about seasons. Day/night and circadian clock. Plants have a bona fide molecular circadian clock, conceptually similar to ours. It's a set of transcription factors, CCAI, HNA, and other in Arabidopsis that form interclocking negative feedback loop. Each protein represents the transition of the other on roughly a 24-hour cycle. I see. The photochrome system entrances this clock to the actual light-dark cycle, the same way lights through your retina and train your suprachiasmatic nucleus. If you put a plant in constant darkness, the clock keeps ticking at roughly 24 hours, but drifts, just like your circadian rhythm does in a cave. Interesting. So circadian rhythm is basically a set of proteins that flip into each other, right? Transcription factors. I'm not getting it. This is too foreign for me. Nucleus. Oh, so that's one protein. The day, the evening, the night. Sorry, inside of nucleus, cytoplasm. Wow, they make proteins to track a time? Or they have concept of time because of proteins. How fast they can do that, right? So basically, if we put enzymes to create those proteins faster, we can squeeze a couple cycles into regular 24 hours. I'm trying to find a way to speed up that cycle. Not sure if it's possible. Okay, seasons. This is really good part. Plants don't measure average temperature dropping or have any concept of a year. That's what I thought, but that's interesting. What most of them actually measure is night length. Yes. That's what I thought too. I thought it would be some kind of length. How long is the day and night? Yes. Totally makes sense. But that means that from the light, the ratio would be shifted, meaning we can trick them to have 2 seasons in 1 season, or aka 2 years of life in 1 year of life, if we speed up the circadian rhythm. This is called photoperiodism, and it was one of the clean discoveries in plant biology in 1920. Nice. Plants fall into categories. I want to see what was the experiment to discover that. Also, can we artificially speed up circadian rhythm? Or length of the night / day to trick plant to switch seasons faster than normal year. It should be new thread, right? Not a reply, a new thread on the Is it Perplexity has this? Like what AI tool have it? Otherwise, it's too much of. Plus, also into categories. Short day plans. Plants fall into categories. Short day plants, which flower when night are long. Rice. Wow. And long day plants. Short day plants and long day plants, which flower when night are short. Wheat, Arabidopsis spinach. The name is historically backwards. That's why I was confused. They really measure night duration, not day duration. Okay. The mechanism, at least in works like this. The circadian clock drives rhythmic expression on a protein called protein accumulates in the late afternoon, but CO is unstable in the dark. It gets degraded. So in short days, CO peaks after sunsets and get destroyed. On long days, CO peaks while there is still light. And light stabilizes it through phytochrome and cryptochrome signaling. Stabilized CO activates transcription of gene called FT, flowering locus T. Wow. And FT protein is mobile florogene signals that travel from the leaf to the shoot apex and trigger flowering. It's a consistence detector. The clock says express CO now, and the light environment say is still light when CO peaks. Only when both conditions align does the plant flowers. Wow, they have compute. That's awesome. Coincident detector, yeah. Temperature does matter, but differently. Some plants winter with many biennials need a prolonged cold period before they can flower. This is called vernalization. But, oh, I see. But not in thermometers in a way you'd think. It's epigenetic. Prolonged cold caused progressive histone modification. I forget what histone is. That's a sugar that's right. That's a sugar around which the DNA is wrapped. And epigenetic can, and the way how tight histone are, you couldn't even read the DNA chain. Yep, makes sense. Especially 3 meter at locus called this is too much word. Weeks of cold gradually silence through chromatin remodeling, and this silencing is methodically stable. Methodically. What is this? Methodically. That's crazy. Methodically.
Methodically.
Methodically. Wow. Methodically. Methodically.
Methodically.
Methodically. I was so off. It persists after the cold is over, so the plant isn't sensing its cold right now, is accumulating an epigenetic record of I have been cold for long enough. Think of it as a right once counter rather than a thermometer. Interesting. So we can trick that one for sure. The experiments you are imagining, they have been done. Awesome. This way it gets fun because these are exactly the kinds of experiments that nail down the mechanism. Night breaks experiment. Take a short day plant that needs a long night to flower. Give it a 16-hour night. It flowers. Nice. Now give it the same 16 hours night but flash it with a few minutes of red light right in the middle. It doesn't flower. The pre-flight pulse resets the P of R pull and breaks the darkness measurements. This proved it measuring continuous dark duration, not cumulative flight. Nice. Honda Hammer experiment. See, everything what I ask already been in that answer. Where is the dashboard? Okay, let's go. Nanda Hamner experiment. These are the ones closer to what you are describing. Researchers give plants light dark cycles of non-24 hours period, like 6 hours light, 6 hours dark. Perfect. 12-hour cycle, 6 light, 18 dark, 12. 30 dark, perfect. Yeah, that's exactly what it is. The result plant showed a rhythmic flowering response that peaked whenever the total cycle length was a multiple of 24 hours. This was definitely evidence that the circadian clock is involved. It's not just a simple hourglass timer measuring darkness. It's a clock interacting with the light signal. Greenhouse trick. Commercial growers exploiting this routinely. Yeah, I would assume, right? That's why they grew stuff like within a few months that's supposed to grow like a couple years. Interesting. What's the word? Oh yeah, I remember it was speech.
Chrysanthemum. Chrysanthemum.
Okay, chrysanthemum growers use blackout curtains to give artificial long nights and force flowering out of season. Right. Right. That's how. Mhm. Poinsettia timing for Christmas is entirely controlled this way. Long day plants can be forced to flower within night break lightning. A few minutes of light in the middle of the night is enough. Wow. So those Christmas plants that they making it, right? That's the one. Oh, my hand is so itchy. Have poison oak all over. Just to make sure it's, yeah, that's the one. To have points ready for Christmas by them was the best selection. The bloom and this plant started strictly of 14/16 hours of daily uninterrupted darkness using a closet or box from October 1 to Thanksgiving to ensure fully colored brightly bright. Wow. That's classic. Accelerate seasons. If you give a vernalization requiring plant short cold pulses interspersed with warm periods instead of continuous cold, it doesn't work. The epigenetic modifier of yes. Yeah. We got that. That's for long-term cold lovers. So directly answer your question, the season of day-night is photochromic and circadian clock, the sensor nervous system of pathway guided by circadian clock, and yes, you can absolutely trick both systems, and the experiment doing exactly that of what proved how they work. If we artificially create dark, yeah, that's what the great question. And thinking through carefully actually reveals why the system is more clever than it first appears. The rate asymmetry is everything. The two directional of PFR, PR can say operate on completely different time scale, and this is what makes your thought experiment interesting. This photochromic reaction, a photon hits the chroma, it isomerizes. It's essentially instantaneous on biological timescales, milliseconds to seconds. Within minutes of turning on full spectrum light, the cell reach a photostationary state of roughly 85%. Oh, okay. So it's basically collects a photon and that ratio drifts into 85 to 15 because sunlight has more red than far red. This thermal reaction. Oh, that's a thermal reaction. No photon involved, just slow, spontaneous confrontation, relaxation. The half-life varies by species, but its half-life varies by species, but typically in the range of 1 to 6 hours. This is the slow side, and the rate limit step for the whole measurement system. And the rate is limiting step for the whole measurement system. So if it 1 hour, interesting. So we can make night only 1 hour for them. If you run 3 hours light, 3 hours darkness, 4 full cycles per 24 hours, during each 3 hour rate prepare snaps to 85, almost immediately, then parks work fine, nothing changed, but during the each 3 hours dark period, which half-life of, let's say, 3-4 hours, you only get through the roughly one half-life of dark reversion, so period drops to maybe 40 to 50. But it sets like temperature, right? So before the light comes back on and hammers the light back to 85. Compare that to a natural 12 hours night, the plants get 3-4 half-lives of dark reversions, and PR drops to maybe 5-10 by dawn. So the ratio does oscillate faster, but with much slower amplitude. Never gets deep enough. The plants for the pool, they PR heavy on average cross whole 24. It took from the spirit almost like continuous light. This is exactly what the night breaks experiments already showed. And this is why it comes back to those experiments I mentioned. Even a single 5 minutes red light pulse in the middle of the 6 Hold on, but what if you dropped? I need to know that. You mentioned that reaction from PR to PFR, it's actually related to the temperature. So if we rapidly decrease the temperature, would that reaction increase and we can actually get to the 5 or 10% of PFR? The second clock can't entrain to 6-hour cycles. Can't entrain to 6 hours. This is the other layer, and the Genius clock has natural period near 24, and it can entrain. Can or cannot? That's closer to 24 hours. Within a few hours, either way, roughly 18 to 30 hours total cycle length, depending on the species, but a 6-hour cycle is way outside the range of entrainment. The clock would essentially ignore the rapid cycling and either free run at its natural 24 period or become arrhythmic. This matters because the photo periods measurement isn't just photochrome's axonal length, it's a coincidence between the clock-driven CO expression peak at the light signal if the clock. Yeah, I remember that. So to answer direct no, you wouldn't trick the plant to experience faster days. You do something more like jamming the signal. System would see something close to constant light, never enough dark to appear, and the circadian clock would decouple from the external simultaneity. The plants would experience compressed season. It would experience something more like sensory confusion. Growth would likely continue, photosynthesis still works, but development, transition like flowering, would be disrupted. The way to actually trick the plant into faster season is to compress the photo periods, change gradually. Start with 16 hours light, slowly shifting to 8 hours. Yes, so basically the night length. Keeping each cycle at 24 hours total so the clock can entrain. That's why the coincidence detector track normally, but the summer to winter transition happens to an accelerated schedule. Maryland mammoth. That's an experiment. This bird grew huge. Was almost accidental. This discovery was almost in 1918. Gardner and Allard were working at the USDA in Maryland with a tobacco mutant called Maryland Mammoth. Huge, 10 plus feet, wow. But refused to flower during the normal growing season. It would only flower in the greenhouse over winter. Every other tobacco variety flowered fine in summer. The obvious variables, temperature, soil, nutrition, were all the same, so something else was controlling it. So the hypothesis is it might be day length. To test this, they I need to fix my arm. They did something simple and clever. They built dark chamber, basically wooden cabins, and moved plants in it and out of them to artificially control how many hours of light the dark plants received each day, independently of the actual season. They give Maryland Mammoth plants artificial short day, long nights during summer by wheeling them into the dark cabinets and afternoon. Yeah. Normally, the plants flowered in the middle of summer when they normally never would. Then they did the reverse. They give other plants species artificial long days during winter using supplemental light. Long day species flowered winter, yes. Can you speed up the circadian clock itself? Yes, actually, in two ways. It just told me that no, it's 18 or 30 hours. Generally, in a mutation in a core clock genetic change in period, the TAC1 mutation lost the function, shortens the clock period from 24 to 21 hours. The ZTL mutant lengthened the various other periods mutants. Thank you. Okay, so genetically you can mute some genes to speed up the clock. Measure better. A plant with 21 hours clock and trained to a 20 hours light dark cycle performs measurably better in terms of growing and carbon fixation than the same mutant forced into 24 hours cycles, which is nice demonstrations that the clock actually matter for fitness, not just flowering. Pharmacological casing can clock proteins and affect period lithium, but pharmacological approach is bluntly so you can change the clock speed, but in practice it's not the most useful lever for what you are asking. The particular answer to your real question is compressing seasons. Yes, and this is actually straightforward. Since the plants measure photo period on a per day basis, and it doesn't have any calendar or memory of how long the current season has lasted, yeah, you basically increase the length of the night and it think it's winter. As aggressive you are. You can compress the season photo period schedule as aggressively as you want as long as you follow two rules. Keep each individual cycle close to 24 hours, right? So the circadian clock can entrain. Change the light-dark ratio gradually enough that the photoperiodism measurement photoperiods measurement system tracks it, but gradually. Here can means shifting by 30-60 minutes per day, which is much faster than the natural rate of 2 minutes per day at temporal latitude. So if natural summer to winter photon change takes 6 months, going from 16 light to And you could compress that into 2-3 weeks by shifting the light period by 30 minutes per day. The plant's clock and trains to 24-hour cycle, so peaks, everything work. The plant transition to winter mode or summer mode on whatever schedule you impose. This is literally what commercial greenhouse operations do. Poinsettia growers start imposing long night conditions in early October to force flowering by December. The one things you can't compress, vernalization. If a plant requires 6 weeks of sustained cold to epigenetically silence, you need to actually give it 6 weeks of cold. That part is cumulative timer, which is a real minimum duration, and you can't speed it up by cycling temperature rapidly. Rather than just cold, but you can compress it from weeks to days. There is great instinct. This when we shift in the ratio, right? Oh, this one is gonna be short. Yeah, I need to go eat. This you're trying to find another lever to drain the PR pool faster, yes, but it actually goes the opposite direction from what you expected. When I said PR dark revision is a thermal reaction, that's chemistry jargon, meaning it's driven by ambient thermal energy, molecular vibration kinetic energy rather than by photons. It doesn't mean heat makes it happen. No, I was saying cold makes it happen. It follows the Arrhenius relationship. Express how chemical reaction rate constraints K depends on temperature, activation energy. Virtually every non-photochemical reaction rate lower temperature means less thermal energy available to cross the activation barrier, which means the reaction runs slower. So if you drop the temperature during your 3 hours dark period, actually reverse PR more slowly. The half-life gets longer, not shorter. Instead of that dark reversion can slow dramatically. So you need 3 hours. Yeah, this happens measure experimentally. It's several decode specific PR dark version. The counterintuitive implication. This means cold winter nights are actually worse at drain than warm summer nights. Yes, on per hour basis. Measuring is still worse because winter nights are so much longer. Got it. If you want to speed period depletion, you actually options are far red light instead of total darkness. Heat the plant with pure far red light. Right, makes sense. This drives far red light. Per convert photochemically fast, seconds to minutes, and completely independent of the temperature. Oh, this is actually what happens at natural dusk. As the sun as the sun drops in the current, the red far right ratio of sunlight shifts heavily towards the far red because red light gets carried more by atmosphere. So PR pulse started raining photochemically before darkness even begin. A brief pulse of far red at the start of your dark previous would crush PFR to many. Do we have PFR in humans, in mammals? By end of the day, far red treatments are a real tool in plant biology experiments. Researchers use them to rapidly establish low PFR condition without waiting hours for them to revert. Okay, perfect. Rise the temperature going the other direction. So very interesting. So you can speed up seasons but not very rapidly. Okay, we'll be back.